we're going to be talking about AP Biology Chapter 8.3, and we have three main objectives that we're going to be covering. The first is naming and describing the three types of different cell work, and then the second is going to be describing ATP as a high-energy molecule, and then the third is describing how ATP hydrolysis does work. So first we're going to be looking at naming and describing the three types of cell work, which are chemical, transport, and mechanical energy. So first let's talk about chemical.
Think of Legos, the building and breaking apart of things. So things like putting together monomers and dehydration reactions.
So yeah, these are basically just all the building up or breaking down processes that the cell does. And like I said, examples of this are dehydration reactions and also hydrolysis. Now let's take a look at transport, which this is basically the pumping and transport of material at the cellular membrane that we talked about last chapter, such as pumping an ion against the concentration gradient or active diffusion. Now let's discuss mechanical energy.
Think of the energy expended when Dr. Lyons, Tommy, and Matthew hit the gym to lift some weights.
So this is any physical movement that the cell does, such as the pumping of cilia and flagella, amoeboid movement, and then also the processes in contracting muscles. So now that we've talked about the three types of cell work, we can talk about energy coupling. And the basic idea behind energy coupling is that the energy from an exergonic reaction is used to drive forward an endergonic reaction. And just to review, an exergonic reaction involves the release of energy from a chemical breakdown, and an endergonic reaction involves the intake or absorption of energy from building something up. We can apply and look at energy coupling in ATP and ADP. And we know that ATP is a high energy molecule, but how do we know this? Let's take a look at structure and function. So here we have a lovely ATP molecule, and here's our nitrogenous base, which is adenosine, and then we also have our ribose sugar, but our really star players, the ones who are going to be doing the most work, are these three phosphate groups. And it's important to note that each of these phosphates actually has a negative charge, and these negative charge all compress next to each other, kind of act like a coiled spring. That's how the textbooks describe it. So these charges next to each other result in an unstable and reactive molecule.
Another example to help this make sense is sticking two magnets with the same charge next to each other, the same poles next to each other. They won't get close to each other because they just want to repel because they're the same charge.
So the phosphates repelling and pushing against each other result in a really unstable molecule, like I said before, which leads us to what we call ATP hydrolysis. And just a reminder, hydrolysis is the addition of water to break something down. And in this case, it's going to be ATP into ADP. So sketching this out, we have our ATP, and we're inserting a water molecule between the second and third phosphate, which results in ADP plus another phosphate plus a bunch of energy. And this energy, the energy that's released from this reaction, is negative 7.3 kilocalories per mole in standard conditions, but in cellular conditions, it's negative 13 kilocalories per mole, which is a lot of energy that drives further reactions in the cell. So going back to our definition of energy coupling, this energy that is released through ATP hydrolysis is used to power other functions in the cell through a phosphorylated intermediate. And this is a big term that I just threw at you, so let's break it down. To make this make sense, we're going to use a really simple example. So let's say we want to add this triangle and bond it to this square in order to create a house shape. And let's say it takes this house 3 kilocalories per mole of energy to make. But when we add the phosphorylated intermediate from our ATP molecule, we get a house more quickly because what we're doing is creating a more reactive square, and that square wants to bond as quickly as it can to the triangle so that it can be in its more stable state of the house. And since the reaction uses 3 kilocalories of energy to build this house, we're really using less than 1 ATP, the energy released from less than 1 ATP to build this house. So bottom line is that the phosphorylated intermediate creates a less stable and more reactive molecule to drive reactions. Another thing that can happen when you phosphorylate a molecule is a shape change. A lot of times with protein pumps in the cell membrane, which the shape change allows them to either dump something into or out of the cell. So the bottom line here is energy given off in sticking a phosphate onto a molecule is that it causes that molecule to do productive work for the cell, and that can be any of the three types of cell work that we discussed at the beginning of this video. So obviously we're using a lot of the energy from ATP hydrolysis reactions to fuel cellular functions, but we need these ATP molecules back so we can basically do it again. So this kind of works in a cycle, and I will sketch that out for us right now. So we start with our ATP molecule, and like we just talked about, we release a phosphate and energy to perform other functions in the cell, and we're left with ADP because we released that phosphate. So to get our ADP back to ATP, we need energy and to add on a phosphate back onto the molecule, and that energy comes from the food that we eat. So that's basically everything from section 8.3. We talked about the 3 different types of cell work, we talked about ATP, the function and structure, and how we use ATP hydrolysis to do work.